Thank you everyone for joining this webinar on time to event for PRO endpoints, avoiding pitfalls for interpretable and valid results. I am Stacie Hudgens. I am the founder and CEO of Clinical Outcome Solutions, and we'll be allowing Jen, Beaumont, and Libby Floden to give more detailed introductions to themselves as they move on to their sections. Clinical Outcome Solutions is a clinical outcome assessment research consultancy. The members of this team today are presenting on behalf of the quantitative science team, specifically in biostatistics. Our combined experience over the years has really focused on quantitative and qualitative research in the COA space. And so what we wanted to do is to bring lessons learned around time to event analyses, particularly focused on oncology, while time to event is used in many other analytic areas. We're going to focus a bit on oncology today. So without further ado, I'm going to get started. Per our earlier presentation, please use the Q&A to post your questions, and we'll get to them as we can. This chat is disabled, otherwise it would be popping up and distracting all of us, so we're going to use the Q&A for that. So the learning objectives for our webinar today are really focused around research questions, the Estimand framework, identifying intercurrent events, and analysis and interpretation. So research questions, so time to event analysis for PROs when it's an appropriate analytic approach. The Estimand framework, the role of the Estimand framework in defining and analyzing the time to event of PROs, along with the challenges to watch out for. Identifying intercurrent events, which are intercurrent events that might impact the time to event analysis of patient-reported outcomes. And then analysis and interpretation, specifically strategies for accounting for intercurrent events, analyzing and interpreting the time to event PRO data, and how to avoid common pitfalls. For the sake of this presentation, we are not focusing on the overall umbrella of clinical outcome assessments. We are focusing on patient-reported outcomes. For some of the other COA assessments, such as observer-reported outcomes or clinician-reported outcomes, you can consider time to event analyses. However, I think you have to put a bit more time and thought into it, specifically to ensure that the research questions are aligned to the analytic strategy. So questions to consider, and this is redacted from Anoda's 2015 article. And we do have and will be providing, when we load the slide deck onto our website, you will have the ability to see all of the bibliographic references, including several of our own, will be made live on that link. So questions, what is the threshold for defining deterioration? Within the context of this presentation, we are going to focus on time to deterioration. You could, again, because of your research question, you could be looking at time to improvement. It's generally indication driven, and in oncology, it could be early stage disease when you're thinking about time to improvement, whereas in late stage disease, you're attempting to delay deterioration. So in this case, we're going to talk about what is the threshold for defining deterioration, how many deteriorated visits are required to meet the definition of an event, what if patients improve after their first deterioration event? What about patients who are missing a baseline score? How do you look at time to deterioration when we don't know where they started? How is time equal to 0 defined? What if there are missing assessments? Libby's going to cover a bit of that, as will Jen. How many assessments do I need? When you're thinking about your schedule of assessments, when you're planning your clinical trial, how many times do you need to collect the PROs? What if patients stop treatment prior to deterioration? What if patients experience disease progression prior to deterioration? How do you consider that in the framework of intercurrent events? And then what about patients with baseline scores that don't leave room for deterioration? So in that particular case, these are the patients who already enter the study at the floor of measurement, which is generally the worst possible health state. So these are questions that you want to consider when you're thinking about time to event analyses, and we're going to do our best today to present to you different scenarios in which you can consider each of these questions. So the research questions, we talk about time to event. Again, we're going to look at time to deterioration. In time to event endpoints, we really consider two parts of endpoints. First, it's very similar to when we're looking at clinical endpoints. Did the event happen, yes or no? So often in oncology, we're looking at deterioration PROs, really looking at it and how it relates then to progression-free survival, for example. So very similarly, we're looking, did the event happen, yes or no? And then what is the time? So the time to event or the time to the last date that the event was known not to have occurred. You're going to hear Libby and Jen talk about censoring, which is the phenomenon where the event does not occur for all the patients. So the time to event cannot be known for all patients. And so what are some of the censoring rules? And one of the pieces we're going to talk about today are what are the different approaches to censoring, because with patient-reported outcomes, it's multifaceted, right? You have other things to think about, death, progression, rather than just the meaningful deterioration. And then Libby is really going to focus on the Estimand, so the precise description of the treatment effect reflecting the clinical question posed by the trial objective. It summarizes at a population level what the outcomes would be in the same patients under different treatment conditions being compared. So Libby is going to spend a bit of time on this and has done quite a bit of work in the area, including publishing one of the first papers on the Estimand framework. Just for the sake of introduction, I've talked about time to event as a supportive analysis in oncology trials. Really to supplement and support the interpretation of overall and progression-free survival, because it allows us to also, very similar to survival endpoints, look at the median time to an event, such as delaying a patient's functional deterioration, and let's say in physical and role function. Time to deterioration is a specific subset of time to event analysis that is looking at the time to deterioration. Now, in this case, time to first deterioration, time to definitive deterioration, we might see time to first deterioration used in advanced disease due to the likelihood of prolonged deterioration, while in the adjuvant setting, time to definitive deterioration may be more suitable because patients may have a deterioration. So let's say in prostate cancer, patients have a deterioration due to a new treatment, but they can improve later. And so we really want to go over some of these examples for defining events. So again, time to first deterioration, in that consideration, deterioration is reversible. Time to definitive deterioration, which is also called confirmed deterioration, it's the time to the first two consecutive deteriorations of at least one meaningful change threshold. And really the way we think about that is that a patient has a threshold of deterioration, say in physical functioning, let's say of 10 points. And then the next PRO visit, they still have that prolonged deterioration. In that consideration, you also might see death can be the second deterioration event. In this particular case, it can also be considered sustained deterioration. Time to definitive deterioration, the second definition of this, the deterioration is not reversible. So in the first definition, it can be reversible. The second one, it may not be. And then a newer definition that's come up is deterioration-free survival. And this may include any of the above events or death. And in that particular case, you're looking at how long the patient is surviving without a meaningful deterioration. And so one of the things I want to add here that is really important is that we consider what is a meaningful event. And so one of the added pieces when you are considering the example of defining events, what is an event in PROs? And in PROs, you're really looking at using and optimizing meaningful change thresholds within patient change from baseline. And those are psychometric techniques for deriving a threshold that would be meaningful to a patient. These are included in the PFDD guidances by the FDA as well as the 2009 PRO guidance. And there's a lot of working groups going on how do you determine that event. So in terms of an endpoint definition, considerations for each of the time to event analyses, time to first deterioration, it's easy to apply, less impacted by discontinuations because you're taking the first time that a patient deteriorates and you're using that as your event. The negative, a patient is categorized as deteriorated regardless of the subsequent assessments. And so you're using that first time and then you're stopping at that. Time to definitive deterioration, it's more robust definition. It helps to avoid temporary deteriorations that rebound. And then in this case, if you're working in the oncology space, an example is where a patient may rebound is in the first 6 cycles of therapy, a patient may have, say, an adverse event of diarrhea, right? Nausea, vomiting, diarrhea, pain. But due to concomitant therapies, the patient may rebound and now the diarrhea and the nausea and vomiting are under control. But if you use time to first deterioration, that patient would already be flagged as deteriorated. Whereas if you use definitive deterioration, you're able to utilize those subsequent events to see if the actual tolerability event resolved itself. The negatives, it will stop at the definitive time point definition, thus limiting the ability to confirm later time points. So the definition is dependent on the research. Just like any other endpoint that we're looking at, you need to start with the end goal. What is the message that we want to deliver at the end? What is the study population? What is your study design? What is your COA instrument included in the study? What you really have to consider with the COA instrument is the directionality of the scale. So if a higher score is a positive outcome or the higher score is a negative outcome, you have to think about all of those things, including how do you derive the meaningful change and what are the anchors that you use to do so. Intercurrent events, which we will be talking about with Libby and Jen, of course, and then the availability of prior data from similar internal studies and or mechanisms of action that may actually help us derive that hypothesis. And then again, and this is not often well articulated in at least in historic studies, and more of the newer published work, we're seeing a bit more detail, but competitor approach to time to deterioration for study design or comparator arm. Ensuring that you really understand the model that was used, because if you use a specific definition of time to event, but your competitor used a different definition of time to event, you're not going to get the same results. And there will be a discussion from Jen on sensitivity analyses when you're developing your analysis plan. Of course, here's an example. This is a published example in hepatocellular carcinoma. In this particular case, we were looking at no difference or better when we were looking at lenvatinib versus sorafenib. And in this example, we included time to symptom worsening, specifically looking at the median time to which EORTC pain scores declined by a meaningful threshold. And as you can see, the results here were 2 versus 1.8 months, respectively. And what is really nice here is when you look at this Kaplan-Meier curve, which Jen will get into later, you can see that it was slightly better for lenvatinib, but there really wasn't a detriment to the patient's pain outcomes compared to the standard at the time, which was sorafenib. And so these type of analyses really help you look at the patient profile. I'm going to end and pass this back to Libby. And what I'd like to say here is that one of the things with PRO analyses that you have to think about, it's not always that you're going for this massive difference or this big difference when you're coming against standard of care and you're a de novo therapy or you're an add-on therapy. Generally, you want to think about the hypothesis, and most often it's no difference or better. You're not creating any additional tolerability or other patient outcomes detriments to the patients compared to what they would have gotten with standard of care. Libby?
Thanks, Stacie. My name's Libby Floden. I'm a senior director here at Clinical Outcomes Solution. I'm a biostatistician, but also work in psychometrics as well. But my methodological interest really lies in applying the Estimand framework to patient-reported outcomes. As such, I've had several presentations and developed workshops on sort of this family of topics. And I sit on a cross-industry working group for PROs in oncology and the Estimand framework. Okay, so I'm going to assume that all of us here have at least some understanding of the ICH E9R1 addendums on the Estimand framework. But, basically, this is a framework meant to help align the research questions with the design, conduct, and analyses. There are 5 main attributes of the Estimand framework, but it's really the intercurrent events, so number 4 here, where things get really interesting, right? So intercurrent events are post-randomization events, so things that happen along sort of the patient journey that alter the relationship between your research question, right, and your outcome, right? So they're essentially things that are getting in the way of this causal pathway. And so while most of these so the Estimand framework also gives us 5 ways to handle intercurrent events, and most of them can be applied in time to deterioration. My example here is in time to deterioration. I'm gonna really apply and look at both the treatment policy Estimand and the composite Estimand, right? So briefly, the treatment policy strategy. The treatment policy strategy is the closest to ITT, right? So this is where we're looking at the effects of treatment sort of regardless of what happens along the way, right? It's the idea is that this is maybe more of a real world estimate. So we're going to use all data from all people randomized. Whereas the composite estimate, the composite strategy is where the intercurrent events that happen, we're going to now just include those into our actual outcome variable, right? So it's a little bit of a convenient way to handle intercurrent events as long as we're sort of careful about the interpretation. And the assumption here is that, well, in time to deterioration, these intercurrent events also represent a deterioration, right? So disease progression, we're going to include that as our deterioration along with our PRO deterioration. And again, just a very brief word on time to event, right? Today, we're going to apply in this example just our most common methods, right? So the Kaplan-Meier estimator for median time to event, right, or time to deterioration in this sense. And we'll look at a hazard ratio from a Cox proportional hazard model, and that's how we are going to compare the 2 treatment arms. All right, so using the Estimand framework, we're going to pay attention to how we define our events and our censoring rules, right? So this is, essentially guiding us how we're going to use the data that we have in our given trial. So we've simulated an example loosely based on sort of a real life oncology trial, or several real life oncology trials, right? So this is unspecified cancer. And as what we often see, the primary endpoint we will consider to be progression-free survival. And a key secondary endpoint is sort of loosely defined as to evaluate and compare improvements in quality of life, right? So, it leaves us some room on how to apply that, right? So, in looking at time to deterioration, we're going to measure disease symptoms from a fake scale, right? The CSAS, Cancer Symptoms Assessment Scale, and to apply time to deterioration, we need to make this essentially continuous or ordinal endpoint. We need to make it binary, right? And we do that by applying a threshold, the meaningful change threshold. Um, I'm not going to talk about how to define that threshold. That's not the focus of this work here, and there's lots of information, and it's always sort of an ongoing developing topic in PROs on how to define, but we're going to assume that we have a nice robust threshold here. But complicating factors include treatment discontinuation due to adverse events, disease progression, and death, right? So these are our ICEs that we're going to consider. The first estimand that I'm going to define will be a treatment policy estimand, and it can be articulated the following way. Right. Does treatment with treatment A delay time to deterioration of disease symptoms or death? I'm going to get to that in a second, as measured by the CSAS compared to treatment with treatment B in patients with cancer, regardless of progression or discontinuation due to AE. Right. And we want to, as always in good form, to also apply sensitivity analyses. So we can see that our primary estimand is using a modified ITT. This is something we sometimes see in PROs. It's not necessarily an endorsement, but a modified ITT will be using patients that have at least a baseline and one post-baseline PRO assessment, right? So that we have at least one measurable determination for them. Our first sensitivity analyses, this will be the same in both my examples. We'll be using all patients. For both those, we're looking at the first symptom deterioration, and our final sensitivity analysis will apply a confirmed deterioration definition. And so let's review our intercurrent events, right? So death, right? For both of these examples, we're going to use the composite strategy for death, right? And this is partly because of sort of a growing consensus that death is a deterioration, it's a negative outcome, and so applying a composite strategy is generally an appropriate way. But for progression and discontinuation due to AEs, we're going to use the treatment policy approach, right? So this is what we have in green here. So that means we are just going to use all the data that are collected post intercurrent event, so after disease progression and after any discontinuation due to AEs, right? And so you can see it, that aspect of the estimand articulated in the green text. So regardless of disease progression or discontinuation due to AE. So I'm not showing results yet here, right? But, and bear with me because I'm going to try and use our interactive writing tool here. But I want to first look at our event rate versus, compared to our censoring rate and our proportions, right? So we can see that about 40 to 50% of patients are experiencing a deterioration event, right? So that's PRO deterioration or death. It's a little bit lower when we are looking at confirmed deterioration, and that makes sense. But now we also want to be looking at our event reasons, right? So we've defined our event as PRO deterioration or death, but it's helpful and important to understand what event reasons are attributing to our estimate. And we can see that death is about 30-something%, and it's higher in our confirmed deterioration, because you have to remember that the confirmed deterioration of the PRO is sort of a harder thing to achieve, if you will. right? But we do see that over 60% of these first two analyses, it's our actual PRO deterioration that is driving our results. Okay, and then now I'm going to step back and look at our second estimand, right? And this could be considered a composite policy estimand, and we're using the composite strategy for all of our intercurrent events. And so the estimand can be articulated in this way. Does treatment with treatment A delay time to deterioration of disease symptoms, disease progression, AE discontinuation, or death, as measured by the CSAS compared to treatment with treatment B in patients with cancer? Right. And I have a similar table with our different strategies for our different ICEs. And so we can see, as written in the green text, that's how we are articulating within the estimand our handling of our intercurrent events. And again, now we have a similar table, right? So we can look at our proportion of events versus censoring, and we have a much higher, right? In this estimate, we have a much higher rate of events. We're in the 70s, above 70s across both treatment arms, across all analyses. And that's because, right, it makes sense because now we're including all these things in the event, not just the PRO deterioration or death. And so now, once again, looking at our reasons and our the distribution across the different reasons in our composite definition, and death is much lower, right? But that's because we're including progression, right? And discontinuation. So we do see that true PRO deterioration is accounting for, a quarter to a third of our events, right, in our estimate, but we have a fairly high amount of disease progression, right? So these are patients where disease progression is occurring before their PRO deterioration. Okay, so now we get to see the results. These are the hazard ratios plotted here. So we can see that all of our hazard ratios, I don't know if you see that my little cursor moving or not, but we can see that they are all around 0.7, right? So this favors treatment A, and looking at our different estimands and our different analyses within each estimand, we're getting consistent results. They are all below what the 95% confidence interval is all below 1. So we have statistically significant results. This is great, right? This is really, ultimately what we want. We want consistency. We want, we're able to be very confident in our results, sort of no matter how you're defining the intercurrent events and the sensitivity analyses, we are seeing a consistent result. So this is very robust. What is different is when we look at median time to deterioration, right? And so we are consistently seeing that treatment A, which are the blue dots, is favored relative to treatment B, right? Sometimes statistically significant, sometimes not, right? We can look for overlapping of the confidence intervals. But our median time to deterioration is much longer, right? We can think about this as being weeks. Is much longer with the treatment policy because we're not considering all these other events, all the intercurrent events as events, right? So it's sort of harder, it takes longer to reach your event versus when you use the composite approach, right? And to me, this really illustrates the importance of articulating a full estimand and having clear interpretation in your results, right? When you are describing this, right? Describing these results, you need to really know that, well, if it's only, 3 weeks or 3 months or something like that, or 5 weeks or 5 months, that's because it is including all these other endpoints in the definition versus not including all the other endpoints. So just to sum it up, in this example, we saw these hazard ratios that were similar across estimands and across all the analyses, but the median time to deterioration was similar within the estimands, but different across the different estimands. Right. So we need to be careful in our interpretation because simply stating a basic research question may not make clear the handling of the intercurrent events, right? Composite strategy, one critique we sometimes hear is that the composite strategy used for progression may mirror progression-free survival. And, I think that that's true, and it is what it is, but I think that illustrates, or the being able to look at the different proportions of the different event reasons makes a lot of sense here. And further, the interpretation for using other strategies like hypothetical, that can be difficult, right? In the hypothetical, you're having to imagine that progression didn't happen. And furthermore, one of the basic assumptions of a Cox model is that censored patients have the same probability of experiencing the intercurrent event, right? If you're censoring, right? If you censor on progression, than those who do not. And that may or may not be a reasonable assumption depending on your analyses. And I think that wraps it up for me. I'm going to hand it over to Jen.
Hi, everyone. I am Jennifer Beaumont. I'm also a biostatistician, like Libby and Stacie. I spent a lot of the early years of my career working with the developers of the FACT and FACET and PROMIS and NIH toolbox measurement systems, both in their development, their validation, as well as their use in clinical research and phase 2 and phase 3 clinical trials. So I am going to cover some of the issues with protocol development and your statistical analysis plan, as well as touch on a little bit of interpretation. Oops, I'm on the wrong window. So first, this says analytic plan considerations, but the first piece is really sort of at the intersection of your analytic plan and your protocol development. And Stacie has brought this up and when she was speaking about the frequency of assessments. If you are planning to analyze a time to deterioration, you really need to think about when that change is most likely to happen in your disease trajectory and for what your expectations are for the treatment trajectory as well. You don't want to have your assessments so spread out that you're missing where those two treatment arms are differentiating on the timing of your endpoint. But you also need to balance this against patient burden and not have so many assessments that you start compromising data quality because your patients are too burdened. And so this is an important thing where you need to think about your analytics in context with how you're designing the assessment schedule for your trial. And to perhaps sound like a broken record as a panel, the most important thing is to really specify your research question as specifically as possible. Think through your hypothesis specifically and work through the Estimand framework attributes that Libby discussed, because after you've done that, your statistical analyses are going to follow very easily right from that. A lot of times, some of these items I'm going to talk about don't get discussed until the SAP is being drafted or until the analyses are being programmed and people are suddenly like, what do I do with this situation? What do I do with this situation? And that really isn't the right time to make those decisions. You really want them to be based on clinical input and expertise so that it's a really informed analytic decision and not just an arbitrary rule of like, this is what we always do with missing data. So you really need to be thinking through these things. So your statistician should be involved in your protocol development and in setting up the research question so that all of these things that they're going to need to specify the analysis plan are incorporated in that research question, including defining the population of interest, what to do with intercurrent events, all of those attributes that Libby highlighted. And then I do want to highlight that with the Estimand framework, we use this term intercurrent event to define anything that might happen post-randomization that could impact your outcome of interest. In the time to event analysis space and literature historically, there's also a phrase called competing events. And a lot of things that we might have thought of as competing events are actually intercurrent events. And so this, in the time to event analysis literature, you see these things
I think we lost Jen.
Do you want to just take over for a moment? Libby?
Sure. Okay, so competing events, right? So she was discussing how they can be, we can now define these as intercurrent events, although there is a modeling framework for competing events, which could be considered. I think generally we would consider a convening event model to be more of a supplementary supportive analysis rather than your main analysis. Oh, Jen, are you back? Do you want to take?
I reset my router this morning and everything, and my connection was fine until I was talking, of course. All right. Thanks, Libby. Yes, so I think Libby probably covered some of the things about different intercurrent events and strategies. We're not going to talk about specific time to event analysis techniques because that is a whole graduate level course on its own. So we are not going to cover those things, but just want you to be aware that there are methods for explicitly modeling the competing events if that is appropriate for your research question. So think about that. And then we'll talk about some of these other issues too on the next slides. So again, how you define your endpoint, as Stacie highlighted, when it comes to deterioration, first deterioration, definitive deterioration, this is really dependent on the trial setting. And again, this is not a decision that should be left for the statistician at the analysis plan writing stage. It's something that needs to be talked about. Oh, did I lose control?
Yeah, the slide didn't advance, so I don't know if you have control or I can take control and do it for you.
Okay, thank you, if you can. Oh, there we go.
Oh,
Ooh,
okay.
I can take back control now. Okay.
Okay.
Oops. Did it advance that time?
You're good.
Okay. All righty. So again, dependent on the trial setting, you really want that clinical input on sort of what is expected for the disease trajectory and expect the effect of treatment to be. This is not just a question for the statistician to decide and say that we always do definitive deterioration. It's really a research question the whole team needs to be involved in. And then the issue of composite endpoints, as Libby touched on, sometimes this can essentially replicate what you're seeing in the progression-free survival. You want to make sure that you're clear on the interpretation. As Libby showed in her table, it can be really important to display which events are contributing to the events in your analysis so that you can see how much your analysis is driven by progression versus death versus actual PRO deterioration. There is an issue where there can be some complications if you have different rates of some of these things. So if you have different rates of progression, that leaves more or less people available to experience the PRO deterioration. So there you might see some differences across your sensitivity analyses when you're looking at that. Depending on your Estimand, you may include off-treatment follow-up data. But the key thing to this, and this is another place where your analytic decisions need to inform how the protocol is designed, you can only do that if that data is collected. And you also need to be prepared for the fact that sometimes, even with the intention of collecting post-treatment discontinuation follow-up, sometimes that data quality is poor. You want to think about that when you're defining what is your primary Estimand, what is your primary analysis versus what is your sensitivity analysis, because you may or may not have that data available. And that's why it's important to think about it, because that can, if it's important for that to be your primary Estimand for research question reasons, you want to make sure that that is clearly emphasized in the protocol design and in the training of sites to make sure that they prioritize collecting that data. And then again, we're not going into specific methods, but I do want to make sure that everyone is aware. Once you've defined your event, the real tricky part is getting the Estimand attributes listed out, defining your event, defining your rules for censoring. Once that is done, the analysis methods are the same thing that everyone is familiar with that you would apply to a general overall survival analysis. So you want to look at Kaplan-Meier curves. You want to, oh, I lost control again. This does not like it.
It didn't advance, so I took control, I advanced, but now you can have it back.
You're going to look at log rank tests to compare the groups. You might be doing hazard ratios to do an overall comparison between groups. I know we've really focused a lot on time to deterioration today, but you can also use these methods for looking at time to improvement or time to symptom resolution or endpoints like that. And in that case, sometimes it is beneficial rather than presenting the survival curve, the curves where you start with the survival at the top and step down as events happen. It can be more intuitive to present cumulative incidence curves where you're sort of accumulating good events. Some other considerations, again, just like what you would do in your overall survival and your progression-free survival analysis, you want to do all the same things. So you want to include your randomization stratification factors so that these analyses are, statistically analogous to your other endpoint analyses. And then these last few points are really focused on different types of data that you're going to want to think about and deal with. And again, a lot of times this is not specified. Sometimes it's something that comes up as a question in programming. Sometimes the question doesn't come up and just something was done. And if you didn't pre-specify it, it's not explicit and you don't actually know what was done in the analyses. So it's important to think about both how you're going to deal with intermittent missing data. So if you have people that are missing assessments here and there, you could be missing deterioration events if they didn't complete the questionnaire at that time. So you need to think about whether you're going to censor before that missing assessment happened, whether you're going to allow for a certain amount of missing data and just assume that the PRO scores remained unchanged between those assessments. And that's a question you need to think about. And then we've also touched on previously the idea of patients missing baseline. So first, are they in your analysis population or not? You may just exclude people that are missing baseline, but that's assuming that your population of interest for your research question and your Estimand is reflected by people who have baseline data. Or you could include them as an event. You could censor them at baseline. None of these are explicitly and universally wrong or correct choices, but they do have assumptions associated with them. So you need to think about whether those assumptions are appropriate and reflective of your research question. Did I go forward? All right, Libby, go for it. One more.
We're on the slide that's sensitivity analysis.
Oh, I don't see that slide. Okay.
Oh, now we're not. All right, let me try again. Sensitivity analysis. Is that what you want? Yep.
So as Libby has touched on in her example, she had several sensitivity analyses. The important thing to think about is that sensitivity analyses, they're part of the Estimand framework, because when we are doing analyses, whether we think about them or not, we are making assumptions about a lot of things, about the structure of the data, about what the missing data means, what it means when people have disease progression and we don't have PRO data after that progression. Whether we spell them out or not, there are assumptions associated with those. And most of those assumptions are not testable. So that's why we do sensitivity analyses, to make sure that our ultimate conclusion is robust across different sets of assumptions, reasonable assumptions. For example, here, if we count progression as a confirmation of worsening, or if you censor patients at time of disease progression, you are making different assumptions that could lead to slightly different conclusions. And so you're going to need to think about, if you see different conclusions based on those different sensitivity analyses, which set of assumptions is perhaps more appropriate. In addition to sensitivity analyses, where you're looking at, for your primary endpoint and your primary Estimand strategy, what is the sensitivity of your conclusion to changes in the assumptions and the endpoint definition? You may also, perhaps in a supplemental PRO SAP, as opposed to in your primary clinical trial SAP, include some supplemental analyses. And so this might be where you use a completely different analytic strategy. Maybe you do actually model competing events as opposed to including something in a composite outcome. You might look at some other endpoints. You might look at individual item level data that was part of a domain score. And you expect these supplemental analyses, you hope, will help support the story that your primary analyses told you. So just to summarize before I hand it back off for Stacie for regulatory piece.
I think we're losing her summary. Stacie, do you want to just take it from here?
Sure, I'll go forward. I'm good, thank you. We'll let Jen come back for the summary piece when she comes back online. So I'm just going to talk a little bit about the regulatory view. And in terms of the regulatory view, it's really interactions that we've had. Recall that I had indicated we've been involved in many new drug applications in oncology, particularly. And I do want to say, first and foremost, that interactions with FDA and EMA have really been great in the past, uh, probably in the past like 5 to 7, 8 years, really around different statistical techniques. So outside of the clinical statistical analysis plan, really having a really strong PRO statistical analysis plan will help you include all of the analytics that you would really need to derive evidence for the totality of, say, efficacy or tolerability evidence to support a new drug application filing. And I think, optimizing those interactions and those different analytic techniques to really help understand the patient-focused outcomes. I can't emphasize it more that it's been a real pleasure to watch both EMA and FDA asking for more analyses outside of the clinical SAP to really help understand and interpret patient outcomes, particularly because the rationale for the selected time to event model, as well as sensitivity analyses, to Libby's point, can result in different time parameters, right? In Libby's example, you have this value to a meaningful event that ranged from 5 months all the way to 10 months with these big confidence intervals, depending on the approach that was used. So specific to patient outcomes, it is really critical to define the event in terms of deriving and justifying the value. But also aligning on what is the value, right? Historically, we've always used time to improvement, right? We've used improvement, meaningful change thresholds, and applied them to every different time to event analysis. However, there's been a big shift. We know now from doing anchor-based analyses and our psychometric analysis that they are not symmetric, right? We know that what is considered a meaningful improvement to a patient is not the absolute value of what would be a detriment. So the example I used earlier around physical function, a 10-point improvement in physical function may not be symmetric to what a deterioration is. So you really have to look at those anchor-based analyses to come up with what is the value for improvement, what's the value for detriment, and sometimes those can be quite different. Recently, the FDA has suggested in a number of publications and presentations the importance of using appropriate statistical methods for analyzing time to event data. Jen talked about this, Kaplan-Meier estimator, and then we presented some others, but it should be evaluated in the context of the overall benefit risk profile of the drug being studied. The sensitivity analyses are incredibly important because we often will also use those to look at, and we have several published examples of this, where we look at say, subsets of patient groups as well. And we apply the exact analyses to each different subgroup. And so I think to Jen and Libby's point, really understanding and defining those and articulating your hypothesis to the agencies, and I'm talking about EMA and FDA, and really putting together, here is my target product profile, here's my research question, here's my approach, here's how I'm handling missing data, here's how I'm handling intercurrent events. Being very specific really helps any reviewer take that into account and review your analytic strategy and provide feedback on what you're attempting to do. So Mallory Fiera, FDA, noted that failing to address these issues in a recent manuscript that's cited here at the planning stage can lead to regulatory delays. I mean, many of us who have been part of previous NDAs, you get the information requests later where you're doing many additional analyses because you didn't pre-specify it at the start. That can cause a delay because now you have to go back and reanalyze all of your data. You could miss an opportunity for a labeling claim, and then the ambiguity and the unclear handling of post-randomization events can be criticized, right? So to Jen and Libby's points, specify, specify, specify. Summary, I'm going to just go over this so that we have 5 minutes for questions if there are any. Treatment timing and setting is a primary consideration of defining time to event endpoints. Specifically, we are talking about in the oncology framework, where you have various stages of disease. You have solid versus liquid tumors, and with all of the add-on therapies and the IOs versus chemotherapies, there's a lot to consider. And now you have all the new device work, especially say in lung around ablation therapy. So you really have to understand, and I really like Jen's point on having cross-functional integration on trying to define this and really honing in, because you can't leave it to the statisticians to decide how do I think about this cancer therapy. In literature, again, there's no consensus on the most appropriate definition of a deterioration event. However, there are a number of guiding references. We have an expansive bibliography in this presentation, but there's a lot more ongoing. To Libby's point, there's a lot of working groups. And then finally, it may be appropriate to assess and evaluate multiple approaches for the event definition and handling of intercurrent events. You can really optimize sensitivity analyses for this. So on that, I am going to come to the Q&A slide. So if anyone has any questions, as Jen said, if you have any, please put them in the Q&A box and we have time for probably 3 questions because we have 3 minutes left. And while we're waiting for questions, I will ask a question first since there's not one. So Jen, I'm going to give one
And
to you.
Stacie, I just want to make a note that if someone wants to, like raise a hand or whatever, we can take their mic off mute. So if they're not typing, we can even just, let them say.
Oh, we have one question here. I'm going to read it out loud. Are you ready? Hi, all. Many thanks for the nice presentation. As far as I understand, there are intercurrent events that are recurring and may affect the hazard of the endpoint of interest. How would we address this issue, like recurrent time to event?
I can start. I think that there are many ways, or at least I can think of several different ways to be thinking about reoccurrent time to events. And really, to echo what Jen said, there isn't necessarily a right or wrong way to handle it, but you have to have clarity, right? Just be clear in what exactly you want to measure, how you're going to define it, and then also carry all those aspects over to the interpretation to make sure you're interpreting it correctly, right? So, if you think about the intercurrent events affecting the hazard, you can composite strategy, you're combining them. Jen, we didn't heavily go into sort of the more complex analytic approach, like modeling competing events, for example. But there are more sophisticated methods that we use and will specify for sort of this supportive analyses to sort of help with that interpretation. I will say that when you get into sort of the more complicated models, they are sometimes based on assumptions that may be hard to justify. There's often not the magic bullet we're always looking for. And sometimes it's best to sort of keep it simple, but, maybe having something more complicated to support and sort of tell the whole story, if you will.
I want to give one example, of one of the more complex models further to Libby. Libby and I tackled this in prostate cancer a number of years ago, and I alluded to this in the intro. Is when you have to think about really thinking about this multiple time to event, as you said, or recurrent time to event. In one area, when patients are asymptomatic and they come into a study asymptomatic, they have a time to a first deterioration, and then they have a time to a second deterioration. And so, to Libby's point, really, and then Jen's as well, putting that together, like, what is the clinical trajectory for this patient, right, on this therapy and in this area. So, for example, in prostate cancer, if you come in asymptomatic, we know that you're going to become symptomatic or have a deterioration in functioning due to now you're taking a treatment that is going to have some tolerability issues. So then we have to think of a second baseline, right? And so I think you really, to Libby's point, you have to just really understand what you're looking at, and not just try and square peg round hole and put the same model on every single um, cancer type and therapy type. So, Jen, did you want to add, or was that?
No,
I think we're
you
at time, but
we're out of time.
we are out of time. I'm just going to let you all know that these will be available. We are going to make these slides available as a link. Our email addresses, hopefully, with us being on this Q&A page, our email addresses are available. You can send us an email and ask for any of our we can share the presentation, we can share any of the bibliography. So please reach out if you have any further questions or you'd like to talk further on this. And we thank you all for attending.